Namaste to all. Welcome to the next session on molecular biology and rDNA technology. In this session, we are going to see about mutation and types of mutation which are taking place in DNA. So, mutation means a sudden change in the genetic makeup is called as mutation. Its results would be immediately visible. In some cases, it cannot, you can say that it is also prolonged and visible after some years. First discovered by Hugo de Veres in evening primrose, that is Oniothera lamarckina, a substance causing generation mutations are called mutagen. Mutation can be classified as spontaneous mutation and induced mutation. So if you take the mutation or mutagen, it is divided into two types. First is chemical based and other one is physical or radiation based. If you take the chemicals, there are different types. But physical, if you take like ionizing and non-ionizing mutagens are there. In chemical, we have base analogs, that is 5-bromouracil, 5-iodoburacil, sorry, and analogs to nitrogen bases, analogs to thiamine. The second type in the chemical is alkylating agents. Third is acridine dyes. And in ionizing radiation, we can say it has X-ray, alpha ray, beta ray, and gamma rays. They damage the genetic makeup by ionizing the atoms present in the molecule. Non-ionizing, if we see, this has UV rays. So let's see about mutations. So in mutation, as we have discussed about the different types of mutagens, based on this, the mutation can also be considered as chromosomal mutation and point or gene mutation. In chromosomal mutation, we have two types. One is morphological mutation and numerical mutation or numeric mutation. In point mutation, we have three types: point mutation, inversion mutation, and frame shift mutation. Morphological mutation can be divided as deletion, duplication, inversion, translocation, reciprocal translocation. In numeric mutation, we have euploidy and aneuploidy. Let's see about the morphological mutations. So the first one in this is deletion. The part of chromosome gets deleted. So what happens here is, consider in a chromosome, we have A, B, C, D on one arm and another arm has E, F, G, H, and it is our centromere. So during the course of replication or during the process, if because of some external force or by intrinsic force, the chromosome has got affected and the certain portion like F and G got deleted, then what happens is there is a change in structure. That is, it becomes as A, B, C, D and E, F only in the chromosome. The second type of mutation is duplication. Duplication means some segment of the chromosome gets duplication or duplicated. So here, again, I am taking the same example of the chromosome as ABCD in one arm and EFGH in another arm. If a portion of the chromosome like EFG is getting duplicated, then the size of the chromosome will increase and it will have a changes in the structure as well as in the morphology. So you can see here it is becoming as EFG, EFGH in the other arm. Inversion mutation, if you see, this is the segment of the chromosome gets cut and gets inverted in the other part. So if ABCD is there and the other portion is EFGHI and this particular portion, that is FGH is getting inverted, the H will come to the F position and F will come to the H position. So what happens here is there will be minor changes because of this mutation And in some cases, it can also lead to major causes for the morphological changes. Let's see about the fourth type in this, that is translocation. If the translocation is taking place, the thing is chromosome piece gets exchanged between the homologous chromosome, that is non-sister chromatids. So consider in this particular diagram, we have the chromosome with ABCD and EFGH, and the other chromosome, which is the non-sister chromatids from which it is getting exchanged, is 1, 2, 3, 4, and 5, 6, 7, 8. So what happens is EFG gets inserted in the other one or exchanged in the homologous chromosome. And it becomes one, the other first chromosome becomes as ABCD and H. So size of the chromosome gets decreased, and the other one will have an increased feature with additional effects, that is of features of E, F, and G. So reciprocal translocation. So in reciprocal translocation, ABCD and EFGH is there. Along with this, if 1, 2, 3, 4, and 5, 6, 7, 8 are there, and they are getting interchanged, then what happens is the ABCD will have 5, 6, 7, and H, and 1, 2, 3, 4 will have E, F, G, and 8. Numeric mutation. In numeric mutation, basically it depends on the numbers. So it is divided into two types, as we have seen, as euploidy and aneuploidy. Euploidy, that is N, that is having one set as haploid, two sets diploid, three set it becomes as triploid, and many is polyploids. So number of changes by a set is euploidy. So if you see the aneuploidy, it is actually the chromosome number changes by numbers. So it can be plus or minus of 1 or plus or minus of 2 in this case. Consider in the one, we can divide this as two cases in which one is deletion and other is addition. If the deletion is there, that is one of the set is getting, or one of the chromosome is changing, that is -1, that is called as monosomy. And if -2, that means nullsomy. If +1, then it is trisomy. And if one more is added, that is +2, that is become as tetrasomy. So if you see the structures of this, in one, only one X will be there and other one is removed. During the packaging or during the mitotic division, during that time, if both of the things, both of the chromosomes are deleted, then it is nullisomy. If we are having three in one of the cell, three chromosomes in one of the cells during the mitotic division, then it is trisomy. If four are there, then it is tetrasomy. So the example of the trisomy is our Down syndrome. So non-disjunction, that is when the chromosome fails to separate during the cell division, this is called as non-disjunction. So if you see this, we are having homologous chromosomes. So during the division, what happens is during the mitotic division, gamete formation in anaphase 1, if you see here, that is both the chromosomes are packaged in one and other one is not having the particular chromosome. So what happens here is, in this particular case, we are going to have the two features in one, that is, and in another one, that is, if this particular egg, if it is considered a female, so egg is there, XX. So, and in another one, it is only one, that is the sperm has to be there. So during the packaging, that is during the zygote formation, what happens is we will have trisomy condition, which leads for the Down syndrome. So point mutation, if you see in the gene mutation category, point mutation, example for the point mutation is sickle cell anemia. So if you see the sickle cell anemia, in the normal hemoglobin, we have valine, histidine, leucine, threonine, proline, glutamate, and glutamate. So 1, 2, 3, 4, 5, and 6 and 7 are there. So in this category, if the sixth protein forming gene is got mutated, then what happens is, because of the point mutation at the sixth position, it can lead for the sickle cell hemoglobin. So it will have valine, histidine, leucine, threonine, and proline. But on the sixth position, they will have another protein, that is valine, and then glutamate. So due to the addition of the valine at the place of glutamate, it will have sickle shaped hemoglobins, which will lead for the anemia. So the sixth position of the beta chain is getting mutated. So glutamate to valine conversion is there. The structure of the hemoglobin changes, that is the oxygen percentages reduces, and change in shape of RBC. So the normal hemoglobin is circular, or sometimes it can be concave. But in this condition, it will become as sickle shaped, that is percentage of hemoglobin is less and percentage of oxygen is decreased. So inversion, if you see the inversion, there are 2 or 3 bases gets flipped by 180 degrees. That is GAG is getting changed from GAG to it becomes as GGA. So 180 degree shift. So it becomes as GGA. And due to this, you can see that in the sixth position, it is we are taking the same example 1, 2, 3, 4, 5, 6, and 7. So sixth position is actually getting changed in this, and due to this only one change is there in the amino acid. So example for this, the next one in this is frame shift mutation. So frame shift is due to the deletion or addition of nitrogenous bases. So consider we are having a sentence like why did the cat eat the rat? So in this, if we are having the deletion or addition, we will see the changes now. So the deletion, if it is taking place at the fourth position, that is a D, then what happens is it becomes as Y, IDT, HEC, ATE, ATT, HER, and AT. If this is like a frame has been shifted to read, so what happens is, since we know that only the triplet codons are read and 3 codons are read at a time, so what happens is, since one deletion of the base is taking place, all the frames are getting shift, and the reading will be also changing, and the factors also changes, that is the protein or the enzymes which has to be produced, it also changes in this particular case. If the addition takes place in this, what happens? So addition of like X is being added in this particular position. So little bit changes are there. That is, you can see here, that is Y, XDI, DTH, ECA, TEA, TTH, and ERA and T. So like this, if one more extra is added here, that also changes the frame to read. So I hope that in this video you have got an idea about the types of mutation and the mutagens, how the mutagens actually affect our cells, and the mutation also actually affects our cells by different methods like chromosomal as well as the gene mutation. So in the next video, we will speak about the spontaneous mutation. See you in the next session. Thank you. Have a good day.